genetic findings in neuropsychiatric disorders or the genetic influences on neuropsychiatric disease risk. And obviously there is a connection between neuropsychiatric disorders and neuroimaging phenotypes. I mean, this is connected, obviously. And it's very interesting, I think, because we have learned a lot from genetic studies in neuropsychiatric diseases, and these experiences help us also to apply them to neuroimaging phenotypes. And I have always the impression it's a little bit like the neuropsychiatric genetics came first and then neuroimaging genetics followed, and uses a lot of the experiences that we made there. So it's very, very valuable. So psychiatric disorders, and actually from my personal history, I started in psychiatric genetics, and then that was more than 20 years ago, and then also moved into the field of neuroimaging genetics like 10 years ago. So, and it's really interesting to have both views. And so this is an overview of psychiatric disorders. They are heritable disorders of the brain, and they are very common in the population. So here is the lifetime prevalence for, I think these are 13 different neuropsychiatric disorders. This is anxiety, so the abbreviations, you can see them. I will give you the reference from that picture here on another slide. This is anxiety disorders, alcoholism, major depression. This is ADHD, bipolar disorder, schizophrenia, Tourette syndrome. And you see they are ordered by the lifetime prevalence, how prevalent they are. Anxiety is obviously very prevalent, about 30% prevalence in the population. Tourette syndrome has about 0.5%, schizophrenia 0.5 to 1%. So, but overall, these are really common disorders in the population. And then obviously, we want to know, is there a genetic contribution to these phenotypes? And you can measure that by measuring twin or family-based heritability. And that has been done, so you can basically measure in families or in twins how often these diseases or disorders occur together in genetically related individuals. And what you see here is you have heritability values that are a little bit different between the disorders, but you can see they are all substantially, there is a substantial genetic contribution to all of these phenotypes. And so we can be hopeful that we identify the genetic the underlying genetic effects that contribute to these diseases. So what are the aims of molecular genetic research in psychiatry? And this has the basic question, basically, they have not changed since many years. So we want to identify disease genes in order to understand the pathophysiology of these diseases. So what is going wrong actually in the brain, by these genetic variants that confer a risk. That also has to do with brain function. So we are learning something about brain dysfunction. So obviously that tells us something about normal brain function. And then we want to learn about epidemiology. So what's the how do these genetic variants that contribute risk to a disorder behave in the population, also in healthy individuals? That are epidemiological aspects. Pharmacology, how do they impact treatment with drugs, for example? And then a very interesting aspect is the evolutionary aspect. Why are these diseases or disorders so frequent in the population? There's obviously a lot of impairment. These patients are really handicapped, and but still these disorders are very, very frequent in the population. Why is that? So is there also maybe an advantage for some of these genetic variants? because we know, I mean, they also occur in healthy individuals, and maybe they give you a certain advantage. So these are all questions that you can tackle at the genetic level. And then the application is, and I will show you some of this later on, we can use those variants to better classify diseases, and we heard some of it. So you have a mixture of clinical diagnoses, and they are often difficult to separate, and you can maybe use genetic information to better classify the diseases. Then obviously people hope that they can use the genetic information to develop new therapeutics, new drugs, and then based on the genetic information, apply precision medicine, so give individual patients the drugs they need and that they will respond to. And maybe if you recognize individuals that have a certain disease risk in a very early stage, you can maybe initiate specific prevention based on an early diagnosis. So these are all hopes that we have in psychiatric genetics. Now, so the research that is done always follows genomics knowledge and also the available technology. And this is a time frame here from the 1990s to now. And in the beginning, psychiatric genetics started with linkage and candidate gene studies. Jason already talked about these technologies. And then in 2001, the Human Genome Project was finished, so we had a reference sequence of the human genome, and that sort of started an enormous technological development. So all of a sudden, people were interested to have a view on the entire genome, yeah? And so, but they knew we don't have the technology to really look at the entire genome. And with that information, with the human genome information, these microarray-based technologies were developed. You've heard from Jason about SNPs and microarrays. These SNPs are placed on microarrays. You can interrogate millions of SNPs across the genome from an individual by using these chips that I've depicted also here. And this technology has been used for GWAS and for CNV, so copy number variant studies. And then obviously, Jason also showed that the next generation sequencing, exome sequencing, genome sequencing, that's been used since about, let's say, 2012, 13. So it all depends on the price, right? And now the prices are really falling for NGS sequencing, exome sequencing, genome sequencing, and this is developing now. So this is also something that Jason has shown already. So the strategies are basically the GWAS strategy to identify common risk variant for the disease, and exome and genome-based sequencing to identify rarer variation with larger effect size. And so let us start with the GWAS. What has GWAS contributed to psychiatric disorders? And when it came up, actually, about 10, 12 years ago, there was great hopes that this would really contribute to complex genetics. And in the beginning, it was a difficult path. So people had these microarrays. They had certain sample sizes with diagnosis of psychiatric disorder and other complex diseases, and they wanted to study them now with the GWAS technology. And one of the first papers was published in Nature in 2007. That was by the Wellcome Trust Case Control Consortium. So a consortium that brought together individuals with different complex diseases and shared control individuals. So they had 2,000 individuals from each diagnosis. And I will show you which diagnosis this was. There was one neuropsychiatric disorder, bipolar disorder, then there was coronary artery disease, Crohn's disease, hypertension, rheumatoid arthritis, type 1 and 2 diabetes. And so these are the so-called what we call now Manhattan plots. So that gives you an overview of the result of a GWAS. And you know that you want to see these sort of skyscrapers that indicates you there is a position in the genome with SNPs that are highly associated with the disease. And if you look at these, if you look at this picture, it's not too impressive, yeah? So we have, for example, for bipolar disorder, there was not one SNP that exceeded this threshold for genome-wide significance. We had for coronary artery disease, there was one hit, yeah? For Crohn's disease, there were more. Hypertension also, no hit. And rheumatoid arthritis, type 1 diabetes, there were a few hits, type 2 diabetes as well. But overall, so it was the first example of GWAS in different complex diseases, but it was sort of disappointing because people had expected now we're going to find really a lot of variation that contributes to these phenotypes, and it was there were not many actually. And so there was a lot of discussion at that time. So are common variants in fact the ones that contribute to these diseases? What is the proportion of, what is the contribution of common variation to these phenotypes at all? Or are there other types of genetic variation that we should take into consideration? It was an enormous discussion at that point, because people, and I have to say, I think that is important because our expectations at that time were still, we have common variation in the genome that has considerable effect sizes, and we should, with a sample size of 2,000 patients and 3,000 controls, we should see a number of these variants popping up. And obviously that was not the case, so some of it also reflected that our expectations were wrong at that time. And this is another example from one neuropsychiatric disorder, schizophrenia. This is a GWAS result from 2009 from the International Schizophrenia Consortium with a considerable number of patients and controls, more than 2,600 cases, 3,300 controls. So, and if you remember the times of candidate association studies, people worked with 100, 200, 300 patients and the same number of controls. So at that time, this sample size was really, really big, yeah? And still, there was nothing. So there was no genome-wide significant hit. And so, and then at that time, there was, I told you this discussion, should we give up? Should we maybe give up on GWAS? And should we do something different? And it was a little bit before that time that a big consortium formed, the Psychiatric Genomics Consortium, which has been really successful over the last 10 years. And within that consortium, I'm also part of that consortium, we said, no, we should not give up on GWAS. Probably we are just overestimating the effect sizes of the variants that are involved in psychiatric diseases. And we just have to increase the sample sizes and find, and by this we will probably find the involved variants. And a little bit later, a larger study came out. So this is now from the Psychiatric GWAS Consortium. People started to put their samples together, and with that, we achieved much higher sample sizes. These were almost 10,000 cases, 12,000 controls. And now we had 5 genome-wide significant sites. And that was really, so it was time for hope. At least now we had 5 really significant hits, and that gave hope that by increasing the sample sizes, we would find additional hits. And that was indeed the case. So 1 year later, there was a new freeze, additional samples included, now 25,000 cases, 28,000 controls, and there were 62 genome-wide significant sites. And 2 years later, that was published in Nature. There was this work reporting 128 genome-wide significant sites with 35,000 cases, 46,000 controls. And the latest freeze now in the PGC Schizophrenia Consortium from 2018 involves more than 65,000 cases and 88,000 controls, and there are now approximately 250 genome-wide significant sites. And that is really an impressive progress, one has to say. Starting from zero, with 2,000 cases and controls, to more than 250 genome-wide significant sites. And so I will come back to this later. So Jason, we talked before this session, and he asked me to also include a view on candidate genes. Jason, you also mentioned that. You said these were the dark times, basically, people looking at candidate gene, making biological hypotheses and trying to associate a genetic variants in candidate gene with the disorder. That was not very successful in the past, 20 years ago, but there were different reasons. The one could be you've just picked the wrong candidate genes, or the other, the sample sizes were not large enough, so the power was not good. And now with the GWAS data that we have, for example in schizophrenia, these huge huge GWAS data sets, we can also look into these candidate gene hypotheses once again. So we have a number of candidate genes that came up in schizophrenia over the last 20 years or so. And there is an interesting publication, Matthew Keller is the last author. They looked to what extent are these historical candidate genes in schizophrenia associated in the new large GWAS data sets. And they did not find evidence that these candidate genes are more associated than non-candidate genes. So basically, among these genes that were picked based on biological hypotheses, obviously they are not stronger associated than any other set of randomly chosen randomly chosen genes that you take from the genome. So it illustrates, obviously, you can make nice biological hypotheses, but in the end, they can be wrong. And so that was the biological hypotheses in earlier genetic studies, like 10, 20 years ago, that always was an important factor. So the genetic results sometimes were not so impressive, but if you had a good biological hypothesis, then it seemed more evident, and this is dangerous, So the more, let's say, unbiased view that we have now, that we are looking in a genome-wide fashion at the genome to identify variation that is really associated with the disorder or with the phenotype, that is, I think, a clearer strategy. And this is something that we have learned from psychiatric genetics, definitely. I mean, there are also examples, like here I'm giving this example of the dopamine D2 receptor gene. This is a gene that is one of the first candidate genes in schizophrenia. And I remember in my own diploma thesis, I worked on dopamine receptor genes in psychiatric diseases, and that was in the 1990s. And here you see in the latest freeze of the PGC, schizophrenia GWAS, the dopamine D2 receptor gene pops up, so it's genome-wide significant now. So one of the earliest candidate gene now proves to be right, But I have to say, we have not, many of the other candidate genes that we chose are obviously wrong, or the effects of these genes are so small that we even need bigger sample sizes to confirm them, So that is a quite interesting view. Now for other psychiatric disorders, and I'm now giving you a view on bipolar disorder, it's a little bit similar, although not as good as in schizophrenia. This is the latest freeze of the PGC Bipolar Consortium with 20,000 patients and 30,000 controls. And there are now 30 genome-wide significant loci, which is also enormous progress. So in particular for bipolar disorders, we have struggled for a long time to find genome-wide significant hits, but now after all, they are there. And an even more difficult example is major depressive disorder. And for a long time, there were no significant hits at all. And the sample sizes had to be increased a lot to find the first to have the first findings now. And you see, this is a sample size of 130,000 cases and 330,000 controls. So this is probably the biggest sample size in the psychiatric genomics consortium that was needed to identify genome-wide significant sites for major depressive disorder, which tells us something about the genetic architecture of these various, in particular major depressive disorder, but also the other psychiatric disorders. Now, what's the lesson learned? We have learned about the nature of common variants that contribute to psychiatric diseases, and the sample sizes needed in complex genetics in general, maybe neuropsychiatric genetics, but in complex genetics in general, and also, also neuroimaging genetics. This is a plot here is the number of cases and the number of genome-wide significant loci for a number of different neuropsychiatric disorders. And what you can see here, that you have this phase that goes until about 5,000, 6,000 patients and controls, where you don't see that much, in genome-wide association studies. So this is a blow-up of this part of the graph here, and you see for these different neuropsychiatric disorders, you have in this range maybe 1, 2, 3 genome-wide significant hits, so you don't see much. But then comes a sort of inflection point, and if you increase the sample numbers further, then you start seeing more and more genome-wide significant hits. And I think that was mentioned by Jason already. Neuropsychiatric disorder don't differ that much from other complex diseases, because that was also discussed for a long time. Are neuropsychiatric genetics from the genetic architecture point of view, are they different than other complex diseases? Obviously they are not. So they behave like height, for example. So, and what we have learned is polygenicity is a common feature of psychiatric disorder. So we have many risk variants that are involved, and many more than we expected like 10, 20 years ago. So we always thought these are like oligogenic diseases where we have dozens, maybe dozens of risk variants that contribute, dozens of common risk variants that contribute to the disease. And obviously these are hundreds, if not thousands, of risk variants that contribute. So there is a high degree of what we call polygenicity. And then the common variants usually have very small effects. And that is also something that we learned, that we had to learn. 10, 20 years ago, we thought we will find, let's say, odds ratios of 1.5 or 2, which means you have a base risk to develop the disease, and if you have that particular variant, your risk is increased, and an odds ratio of 1.5 would mean your basic risk is increased by 50% to develop the disease if you have that particular variant. And so that would be if you have a basic risk of 1%, such as in schizophrenia, a lifetime risk of 1% to develop the disease. Now, if you have that particular variant with an odds ratio of 1.5, your risk would be 1.5. Your lifetime risk would be 1.5%. So, but we have learned odds ratios are smaller than 1.2 in the GWAS. So the effect sizes really were overestimated, and that's what Jason mentioned already, were really overestimated in the past. Now, we can now take the genome-wide information that we have from GWAS studies and can do interesting things with them. So one is we can try to judge how heterogeneous are diseases. Heterogeneity is a term that reflects you have many different genes that can contribute to a disease, and maybe one patient has gene set A that causes the disease in that patient. Patient B has a different gene set. They can overlap a little bit, but they can also be completely different. And the more heterogeneity you have within a disease, the more difficult it is to find the genetic causes by, for example, GWAS studies. And you need even more sample sizes, the more pronounced the heterogeneity is. And you can do, if you take the GWAS findings that you have, the SNPs that you found for one particular disease, and compare these disease SNPs with other data sets, so you can do a sort of SNP correlation, then you see how heterogeneous these disorders are. For schizophrenia, for example, they seem to be quite homogeneous. So the SNP correlation between different data sets in schizophrenia is more than 80%. So, which is good, and it's probably one of the reasons why we have so good findings with schizophrenia in particular. In ADHD, for example, it's a little bit less, but still very good. Bipolar disorder, also that range. But major depression, for example, you really see there is a drop in SNP correlation, which reflects major depression is more heterogeneous than, for example, schizophrenia or ADHD or bipolar disorder. And then you have this other view that you can have with SNP correlation. What's the correlation between disorders? So to what extent do schizophrenia SNPs also contribute to bipolar disorder? And that is quite interesting. In particular, for schizophrenia bipolar disorder, the overlap is really strong. More than 60% of the SNPs are overlap between these two conditions. And that was something that we did not expect like 10, 20 years ago. So really something that we have learned from these GWAS. And bipolar disorder, major depression, for example, you always thought bipolar and depression, they are closer together maybe. But from the SNP point of view, the correlation is smaller than between schizophrenia and bipolar disorder, which is quite interesting. So you can compare all different sorts of neuropsychiatric disorders and can see how much do the common variants contribute to different diseases, which is an extremely exciting view. And then you can also have a look at different ethnicities, because that is also a big question: to what extent do common variants that contribute to schizophrenia in European descent patients are the same as in African American patients. And as you can see here, for schizophrenia that has been done between Europeans, African Americans, there is a correlation of about 60–70%. So there clearly are ethnic differences, but there's also a strong overlap. It's not like completely separated. Yeah. So the insights basically are, there are different degrees of heterogeneity that we observe. Yeah, schizophrenia, bipolar disorder. So schizophrenia is probably the most homogeneous, bipolar disorder less homogeneous, but really clearly heterogeneous is major depression. Then there is a strong genetic overlap. I mentioned that, and that overlap is particularly strong between schizophrenia and bipolar disorder. It's even stronger than between bipolar and major depression. And it's even stronger, that is also interesting. So the genetic overlap between schizophrenia and bipolar disorder is stronger than the genetic overlap between different sets of major depressive samples. So that is really interesting, I think. The majority of common risk factors are shared between ethnicities, although there are some differences. Now, another view is, another term is SNP-based heritability. So, of course, I have showed you in the beginning, and here's the referent now, Geschwind and Flint in Science 2015, an extremely, really a great paper I can recommend you. So we have looked at the lifetime prevalence of different psychiatric disorders, and we have looked at the heritability for these disorders, and now we can calculate, using SNP-based heritability, what is the contribution of common variation to the overall heritability. And you can see that is very different. So, for example, in bipolar disorder, you have between 20 and 40% SNP-based heritability. In Tourette syndrome, for example, it's very high. It's almost 60%. Yeah. So there are differences, what the contribution of common variation to that disorder is actually. And that is not, it's not the same as the heritability. You can see, this is not basically the same picture as the overall heritability. So common variants contribute more to one disorder than to another disorder. And the rest of the genetic contribution, that is of course the question, and we will come to this later. What is the rest of the contribution apart from the common variation? And yeah, Jason mentioned that we have rare variants in the genome, and they also contribute, or that is the expectation, they also contribute to the heritability. We have common variants, we have rare variants, and of course rare variation has been studied also in psychiatric disorders, and the first that we studied was basically copy number variants, and larger, in particular larger copy number variants, because that was something that we could analyze from the microarray data. And this is a picture of a microarray that is used, a SNP array that is used for GWAS, right? So, and this shows you the signals across chromosome 1. And you see here the SNP signals, and usually they have a certain height. If you have two alleles, then you see a certain signal intensity. And then there may be positions in the genome, like here on chromosome 1q. This is a certain area on chromosome 1q, where you have a drop in signal intensity over many consecutive SNPs. Yeah? And that indicates in this particular position there is a deletion, a microdeletion. And this was a deletion that we identified in schizophrenia a while ago. It's 10 years ago now. So we found that this is a rare microdeletion that is associated with schizophrenia. So it's much higher. It's about 10-fold higher in schizophrenia patients than in normal controls. And the odds ratio is quite impressive. It's 14-15, so which is much higher, obviously, than the usual odds ratios observed for common variation. So this is an example of rare variation, and by looking at these microarrays, you can look at the large copy number variance event, but they have to have a certain size. So there's much more structural variation in the human genome, but the smaller you will not pick up with microarrays, because then it becomes too fuzzy. So let's say if you want to look for microdeletions that are 1,000 or 2,000 bases large, you will not see that in microarray data reliably. Yeah. So for that we need sequencing data, and also in sequencing data it's not easy to recognize them, but we are working on that, on bioinformatic methods to pull out the smaller structural variants from sequencing data. Now, in particular in schizophrenia, we have found many rare CNVs that contribute to schizophrenia, and this is here, these are all the CNVs, deletions or duplications that were identified with the respective population frequency and the odds ratio observed. And there are some, like the 22q11 deletion, that is probably the strongest risk factor, genetic risk factor in schizophrenia, with an odds ratio of more than 100. So if you have that, then you have really a high risk of developing schizophrenia. But what we also saw is that most of these CNVs are associated with different brain diseases. So it's not only associated with one disorder, but with different disorders or phenotypes. And that is an effect that is called pleiotropy. And so that means a genetic variant contributes to different phenotypes. Now, yeah, what is the contribution of CNVs if we look now at these different psychiatric disorders? I have to say we have good evidence that CNVs or structural variation contributes to schizophrenia and autism spectrum disorder, but not that much evidence for other neuropsychiatric diseases. That also has to do with the fact that the sample sizes that can be investigated are particularly large for schizophrenia autism spectrum disorder. Yeah, maybe for bipolar disorder, we also have large sample sizes, and there we see the effect of structural variation is not as large, obviously, than in schizophrenia or in autism. But for other diseases, we cannot tell at the moment because sample sizes are still too small. But it looks there are differences, as if there are differences in the contribution of CNVs to different psychiatric disorders. Now, the contribution of rare variation at the single nucleotide level, so that's what we find when we do genome or exome sequencing, that is still work in progress. And there are some preliminary findings, in particular in autism spectrum disorder or in schizophrenia, showing that there are rare inherited or de novo. So de novo are mutations that occur newly in an individual, so that are not inherited from the parents. So de novo mutations associated in particular with schizophrenia, but they are not well replicated yet. So we have to wait until we make final conclusions here. There are various challenges when we look at rare variants. Statistical power is low, yeah. So we need even bigger sample sizes for the investigation of rare variants than for common variants. And for common variants, you have seen what sample sizes we have now to be successful. So they have to be at least, I mean, it depends on the genetic effect size. If you have really rare variants that have a strong effect size, give you odds ratios of like 10, 20, 30, then you would also see them in smaller sample sizes, but there will be a lot of medium effect sizes that we only see in large samples. Then if you want to look at de novo variants, you need parents. That is also a limiting factor sometimes because you have to recruit the parents, you have to sequence them, which costs a lot of money. Then you have a relatively large amount of private mutations. So each of us carries 100 to 200 private mutations that only we have at the single base level, yeah? And you cannot tell. I mean, if you have something, you can always say one of these 100 or 200 is responsible for your phenotype, but never, you don't know. You cannot really follow it up. And then the functional consequences of rare variants are largely unclear. That is also the case for common variants to a large extent, but it's even more difficult to find the functional consequences and investigate them for rare variants. So, but there are also several large international consortia, and they also look at rare variants now at sequencing data, genome exome sequencing data. So I think results can be expected soon. So that's sort of a magical word also, collaboration, and I think also in neuroimaging genetics, we will hear that later. That's obviously a major factor for success to collaborate to form these large international consortia. Now, something that we can use to interpret the genome-wide findings that we have, you get a huge number of findings, yeah? If you do sequencing, you have many, many rare variants that might be associated with the disease. You have many common risk variants that might be affected, that might be involved in the disease. And how do you recognize what is going on at the biological level? And there you can look at biological networks or also biological pathways that are predefined. So you know there are some genes that work together in biological pathways or in biological networks. And then you can see whether genetic variants that you have identified either from sequencing studies, so here are sequencing studies in families, or sequencing studies from cases and controls, or GWAS study, and you can see whether all these variants, whether they are enriched in certain biological networks. And here's an example for, I think this is for autism spectrum disorders, where you find all your variants, common and rare variants, enriched in certain biological networks. And that also helps you, apart from all the individual associations that you may have, to have a view on the big picture. So what are the biological processes that are involved in the disease? And so biological network analysis with your genetic data, that really helps you to make sense in a biological way. And a further important use of genetic results is, of course, the clinical reclassification of psychiatric disorders. You may know that, but psychiatric disorders are very difficult from a clinical point of view. When we make diagnosis, or a psychiatrist makes diagnosis with a standardized manual, you have a diagnosis for bipolar disorder, but there are features sometimes that look more like schizophrenia. So you have many patients that fall between classical diagnoses, in between, yeah? So you don't really know, is this a bipolar case? Is it a schizophrenia case? There are many examples of patients that, for example, were first diagnosed with bipolar disorder, then 10 years later, psychiatrists say, oh, we think it's rather schizophrenia. And so you have sort of a continuum between these clinical diagnoses, and that also reflects the complex genetic architecture. You have a certain composition of risk variants in individuals, and you have different genetic constellations. And I think you cannot expect that you have just these really separate categories that you can really distinguish from another. So there must be intermediate forms, and so it's just a logical. It's just logical from the genetic findings that we have. And now this is a nice picture, I think, showing the genetic composition of different psychiatric diseases. Here, for example, ADHD, this is autism spectrum disorder, schizophrenia, bipolar disorder, and you see all these circles here with different colors. These are genetic or environmental factors. It's basically both, yeah, taken together. And you see there are many different factors contributing to one disorder. There is overlap, such as here between schizophrenia, bipolar disorder, or bipolar and major depression, and you see what's different and where's an overlap. And so you have a really complex picture of genetic variants and environmental factors all contributing to the phenotype in the end in one individual. And you can look at, for example, what you can do is define the individual genetic risk now with our genetic data that we have in an individual, and try to find out what's the most likely diagnosis an individual has based on the genetic factors. Another thing that you can do is sort of reverse phenotyping using single associated variants or a cumulative polygenic risk score. So now we know, for example, schizophrenia, we have more than by now we have 250 common variants, and you can have a look how many of these 250 risk alleles are present in an individual, yeah? And you can of course you can distinguish between individuals with a high load of these risk variants and a low load, and then you can compare these groups, and you may see clinical differences, and that may help you also for your clinical work, because then you will may be able to see in a few years' time that an individual that carries these particular risk variants is more likely to develop a schizophrenia form in this or that direction. Yeah, so that is, I think, very helpful. Though these polygenic risk scores are also, I think, a very interesting tool to dissect at the clinical level these neuropsychiatric disorders. Yeah, and then of course we need genetic studies to follow up these findings, and I mean they are the first step. We identify at a genetic level pretty unbiased variants that are associated with disease, be it common or rare, but then we need functional studies to confirm them, to make biological sense of them. So what we can do is we can look at animal models, bring these variants into animals and see how they behave, or induce pluripotent stem cells, look at the phenotype at a cellular level, and then we will later hear about expressed quantitative trait loci or methylation quantitative trait loci. That is also something, to what extent do these variants that I have identified, in particular if they are not encoding regions, affect the expression or methylation of the DNA. And then of course neuroimaging studies are a way to follow up these genetic findings, because you can really look into the brain, what's going on if I'm a carrier of that particular variant. And this is only one example of an earlier study that we have done together with Andreas Meyer-Lindenberg of a variant in a zinc finger protein coding gene, ZNF804A, that is associated with schizophrenia. And we have tried to find out if there's a SNP in that gene that is associated with schizophrenia, what is the effect of that particular variant if I do a functional imaging study. And so the results suggested that we have an alteration of connectivity in the dorsal prefrontal cortex, and that was something that was already observed to be the case in schizophrenia patients before. So this is a study from healthy individuals. Yeah. So in healthy individuals, you see this alteration of connectivity if the healthy individuals are carriers of this risk variant. And so these may be studies, interesting studies, neuroimaging studies, to follow up the genetic findings that we have. Just to show you this overall picture, I mean, we have multiple levels and scales that lie between the genomic information, the heritable information, and the clinical phenotype. And so we are in genetics, we are working at that level. But obviously there are multiple layers in between, and Jason has mentioned these different endophenotypes, the endophenotype concept, that we can look what is the effect of genetic factors on the levels in between the genome and the clinical phenotype. And I think this will also help us. Maybe this step is too big, so we need intermediate steps in between. Yeah, and this is my concluding slide. What are the lessons that we learned? So the results that we have in psychiatric genetics is they are most consistent with the polygenic contribution of common and rare variants. Yeah, this is really something that we have learned from these genome-wide studies. The genetic architecture is different between different neuropsychiatric disorders, and we now have the means to see how different they are. And there are some disorders that show significant genetic overlap, something that we did not know before. Yeah, we can quantify the genetic overlap between different phenotypes or disorders. And and the genetic architecture of many neuroimaging phenotypes will be similar, and Tom will elaborate on that further in the next talk. And I don't have questions, but I have put some important terms here that you maybe should remember. There is one, heterogeneity. This is an important term, that you know what heterogeneity is. So there's different composition of genetic factors in individuals, maybe showing the same clinical phenotype. Yeah, and then polygenicity. So the effect of many variants altogether make up the genetic effect for a certain disorder or phenotype. Pleiotropy, the different effects that one genetic variant can have on different phenotypes. Then I've showed you SNP correlation and SNP-based heritability, very useful tools to investigate the relationship between diagnoses, between different phenotypes, and also to estimate what's the overall effect of certain genetic variants on the overall phenotype or on the heritability. And with this, I would like to stop here and thank you for your attention. Maybe you have questions. Sorry, we're a bit behind. I think it's. I think they are very useful because, I mean, after all, you have a cumulative effect, and you know that we used individual genetic variants, and Jason nicely showed it. The individual contribution of one variant, or the what it explains from the overall variance, is much less than 1%. And if you take a cumulative measurement like the polygenic risk, you have a higher, you have a figure that captures a higher proportion of the overall genetic risk. So I don't know what the overall contribution to the variance is, but maybe it's 1%, 2%, 3%, something like that. But it's better than individual variance. That's what I would say. So it's really a progress, I would say. Good. Yeah. That's a very valid question. Actually, yeah, I have shown you that that there is a strong, obviously, there's a strong overlap between genetic risk factors contributing to European schizophrenia sample and Chinese. I think it was African American. Sorry, but there are studies like what's the name of the Converge study that was published 3 years ago, and that is that was a GWAS in schizophrenia with European descent and Asian samples, and you should maybe look into that, yeah. And I think they also made a statement there with regard to the contribution of individual genetic factors to each of these populations. But you are right. There is certainly a risk if you take polygenic risk scores based on European ancestry samples in Chinese, so you may lose something, yeah. But on the other hand, there is this overlap of, let's say, 60-70%. So there is some dilution, obviously, but still, I mean, if you have nothing else to start with, I would start with the polygenic risk score from European descent and try it. I mean, it will be better. You would expect it will become better if you have a polygenic risk score that you can derive from Chinese populations. But in the absence of that, I would start with the European. But look at the Converge study. I think there is information given on risk factors in the Chinese population. Yeah, yeah.